Hi, this is Dr. Yunlong Liu again. This module, we will start to talk about the data visualization. And in this little video, I'm going to discuss the overview of data visualization. So what to visualize? That's the question. I'm sure that at this point that you know that you are going to do a lot of NGS type of experiment. It will generate a lot of data. And normally, you give your samples to the core facility and then pass the data to the implementation. And they will do the analysis and say, you got Excel file. You say, oh, this gene goes this much. Here, identify a peak and things like that. But is there any way that you can really look at your data by seeing, oh, yes, this is something that I detect I should be seeing? So I'm listing a few sample questions here. For example, if I did the DNA sequencing data analysis, can I really see the variants? Not only the VCF files, but see the variants using my own eye. And I can decide whether I trust it or not. If I did a ChIP-seq or a TAC-seq experiment, can I see the peaks identified? And can I see the DNA methylation site? Can I see my RNA sequencing, the rates that cross splicing junctions? We talked about those theories before, but we want to visualize those. In addition to those real data, and I also want to know a little bit more about my specific examples. If I identify a peak here, where the peak is that in the gene region, in the promoter region, the enhancer region, or is this region being conserved? Or what other things do I know about this region? Has anybody else identified this region as well? So all this information are so essential to your data interpretation. It would be really nice to have some way to visualize that. That is something that we want to really cover from this particular video series. Let me give you a few examples. So this is one of the data that we generated in the sequencing core. Initially, I was very, I would say, not confident on what we got because the data is pretty difficult it's a pretty difficult experiment. And this is coming from the FFP samples. We know those samples are very much degraded, sometimes stored for many years, sometimes even decades. And we were able to extract the RNA out. We can do the RNA sequencing analysis. Well, we tried different protocols. Sometimes it's good, sometimes it's bad. But when I got the data, I really doesn't have an idea. Last week, that Dr. Jun Wan and gave an example of looking at summary statistics, and that is very, very informative. But is there any way I can look at my data? So for one of the experiments that we decided to load this into a genome browser, and this is a visualization tool that we will cover a little bit in this lecture series, which is called the Integrative Genomic Viewer. As you can see from here, that this is a gene region, and those are bigger bars, or those are exons. And then this is the signal that we detected from the RNA sequencing. So you can see there is a strong enrichment of the signal that around the exonic regions. That is fantastic. That's exactly something I want to look at. And also, I will see that there are so many arrays that they have different splicing junctions, which means we also sequence the messenger RNA part that intron has been spliced out. Well, this is not adequate to do the analysis, but at least by looking at this, I will have a much different opinion on the quality of the data. This is another example of a TAC-seq. You probably will see similar signals of ChIP-seq. And in the later lectures, we will talk about a TAC-seq. But the point is, this is the assay that help you to measure the chromatin accessibility. As you can see from here, is we see so many different peaks regions, and they are all, most of them are in the beginning of the gene, and which are the promoter regions we know, and there are some of the regions that in the intergenic regions there are possibly to be enhanced regions. So at the very least, you want to load your data into the genome browser or similar tools to see whether you're identifying the correct signal in the positions you expect. And then we will trust a little bit more about the signal regions that we have never seen before. And then for this genome browser, I can further zoom in. So you can see in this particular gene, in the control and treatment conditions, the chromatin accessibility is quite different. I mean, yes, your statistician is going to give you the number to say this gene changed, but it would be so nice to take a look at this and also include something like this in your publications. Further zoom in, and we can see, oh, control and treatment, they are pretty similar except for the middle part. And then if you think about it deeper, this is ataxic signals. And then actually in this region, there could be initially some of the transcription factors are protecting this so that the TN5 ratio transposes cannot be able to cut in. So we get into the biology part in the future lectures, but the point is using the genome browser type of visualization, you will be able to appreciate your data a little bit more. Here's another example of the DNA methylation. So this is bisulfite sequencing treated DNA. We know that after bisulfite treatment, the unmethylated C is going to be converted to T. If you look at this, so you can see all the Cs or most of the Cs are being converted to T. Well, but if you look at this particular location at the CG dinucleotide, this C location, so you can see that most of the C actually are retained as C, and only a very, very small proportion are being converted. This tells you this loci, the DNA has methylation are protecting. And this is the same type of principle, but this is on the reverse strand of the DNA. So you can see this is a CG, and on the reverse strand, this is C, this is G, and the C are being converted to T, and which on the reverse strand will be A here. So you can see that half of the molecules are being converted, the other half are not. So you can have appreciation of your DNA methylation levels at this particular loci. Again, all these examples just give you, we hope that we can have a tool that you can use to load your data into this type of visualization tools, and eventually you can see your data use your own eye. So what is a genome browser? And there are actually several different types of genome browsers. And I'm listing here many open source and public resources are there, and that they can put the genome data and additional annotation data together. So we'll give you some examples. And also that genome browser can provide the contact to organize your data based on genomic locations, as well as help you to search or navigate the areas you are interested in. So if I want to look at the BRCA1 gene, what's going on there, and you have the ability to go there and to take a look. There are many different types of genome browsers. So I'm listing here a few of the most commonly used ones, including the Washoe epigenome browsers, integrated genomic viewers, and the UCSC genome browser. And we will start to generate some of the videos so you can take a look at how we're using it. But actually for this particular lecture, I decided to cheat a little bit, because when I go through the YouTube, there are so many nice tutorials that are being generated by these developer teams. So instead of recording a lot of lectures that how we navigate, we'll still do that a little bit, but I'm going to put in a little more in the playlist about the videos they have generated. I think they are in very good quality, will give you some good idea how these things are going to be used. Just a little bit more information. So in addition to the genome browser that we're used to to see, like the linear version. There are actually circular genome browser available. I'm sure you have seen this in your in different publications. The nice part of it is it not only show you how the signals distributed, and it also have the capacity to tell you that the interactions among different chromosome regions on the genome from your data. Well, that's all I want to talk about today in this particular short video, the general introduction, the overview of the Genome Browser. So we will record a little bit more about how we use this, but at the same time, we'll put in more in the playlist for the existing Genome Browser videos. I hope you enjoyed, and thank you very much. I will see you this coming Friday.